Hello, everyone. Welcome to the Neuroscience Bridge plenary session. I am Vincenzo Bonifati from the Netherlands, and I will be chairing this session together with Etienne Hirsch from France. In this session, we recognize two very prominent neuroscientists who have performed groundbreaking research on topics of broad interest and high relevance for the advancement of knowledge of the nervous system in physiology and pathology. We are delighted that Dr. Madeleine Lancaster and Dr. Don Cleveland have accepted our invitation to deliver the Neuroscience Bridge lectures this year. In a few minutes, Etienne Hirsch will introduce Dr. Lancaster, and I will then introduce Dr. Cleveland. But before that, let me provide you with some important housekeeping messages. The materials presented in this Congress are for educational purposes only and are not intended to be recorded or reproduced. Photography of your screen or any presented content is not allowed, and all relevant faculty disclosure for accredited scientific sessions can be found online in the Congress website. Please post your questions for the faculty in the chat feature located on this screen, and stay for the final 30 minutes of this session for a live discussion and question and answers. And finally, please take a moment to provide feedback in the session evaluation. You can find the link in the chat feature or in the virtual Congress platform. Thank you. We hope you will enjoy this session, and I now give the word to Etienne Hirsch for the introduction of Madeleine Lancaster.
Thank you, Vincenzo. It is now my great pleasure to introduce Madeleine Lancaster. Dr. Lancaster started her study at Occidental College in Los Angeles from 2000 to 2004, where she studied biochemistry. She then went on to complete a PhD with Joseph Gleason at the University of California, San Diego in 2010. After this initial study, she moved to Europe and did her postdoc work in the lab of Jürgen Knoblich at the Institute of Molecular Biotechnology in Vienna in Austria. She then moved to UK, where she is a group leader in the cell biology division at the Medical Research Council Laboratory of Molecular Biology at the Cambridge Biomedical Campus in UK. Dr. Lancaster is a pioneer in the field of stem cell biology and was the first scientist to develop cerebral organoids as a model to study human brain development and disease. Her seminal breakthrough as a postdoc fellow inspired others and opened up a new area of research. Dr. Lancaster's discovery fundamentally changed how we model brain diseases and has the potential to revolutionize the drug discovery process, for example, by developing choroid plexus organoids to model the blood-brain barrier. Dr. Lancaster is the author of publications in the most prestigious journals, including Science, Nature, Nature Neuroscience, and many other journals of the Nature series. Dr. Lancaster has published findings that demonstrate her combined knowledge of development, neuroscience, bioengineering, and application of new technology to generate more mature brain organoid models with impressive properties of long-distance connectivity. And this is typically what we are looking for in the bridge session of our Congress. Today, the talk of Dr. Lancaster is entitled Investigating Human Brain Development and Function Through the Lens of Cerebral Organoids. Dr. Lancaster, the floor is yours.
Hi, it's really a pleasure to be able to present my work at this fantastic meeting. Thank you for the invitation. So today I'm going to be telling you about our work investigating human brain development and function through the lens of cerebral organoids. So in my lab, we're really interested in the human brain and understanding how it gets to be so incredibly complex and enables our higher cognitive capabilities. And we're interested in this not just because it's a very interesting question, but also because a better understanding of the brain is really needed for developing more drugs in the area of neuroscience. And actually, neuroscience as a therapeutic area has really suffered probably more than any other area from a pretty big failure rate in new drugs. And yet it's an area where we really need new therapies because it's incredibly common for patients to experience different forms of neurological and mental illness. So for example, in the UK, 1 in 4 people actually experience mental illness every year. And despite this really high prevalence, the area of mental health research only receives a fraction of the funding that areas like cancer research receive. And on top of this, there's been major issues in the in development of new clinical trials, where a number of pharmaceutical companies have really dropped out of neuroscience because of what some people have called the clinical trial cliff, which is where a number of drugs show a lot of promise in preclinical studies, but then when they get into clinical trials, unfortunately, they fail. And so we think that this is probably due to a combination of factors, but one major one is the fact that our brains are really quite different than those of the animal models being used in preclinical studies. And so what we really need is kind of a human model system. And because we obviously can't do this in vivo, we turn in vitro. And so we've developed this method called cerebral organoids. And these are basically a method to enable us to model the early stages of embryonic development leading to the formation of the developing brain. So to explain this method, I really need to first give you just sort of a brief overview of the key stages of early development that we're trying to model. So after fertilization, the embryonic blastocyst forms, and the inner cell mass here is what will give rise to the entire embryonic body plan. And these cells will undergo a series of morphogenetic movements and changes in identity that will form the gastrula with its 3 germ layers, the ectoderm, the mesoderm, and the endoderm. And the neuroectoderm will form from the midline of this ectodermal tissue and will fold up and close to form the neural tube. And the more anterior portion of the neural tube will then expand and form the developing human brain. And so cerebral organoids are essentially trying to model this process and allow the cells to undergo these self-organizing events with as minimal extrinsic perturbation as possible. So we start with pluripotent stem cells, which can be induced pluripotent stem cells or embryonic stem cells. Embryonic stem cells actually taken from this inner cell mass. And then we just essentially lift those cells off and reaggregate them to form what are called embryoid bodies. And they're called embryoid bodies because the cells start to spontaneously differentiate into the different germ layers in a pretty disorganized fashion, but nonetheless, the cell identities are there. And then, interestingly, there starts to be some self-organization here in terms of positioning, where the neuroectoderm tends to form around the outside. And then that neuroectoderm, when this tissue is placed in a droplet of what's called matrigel, which is a really protein-rich extracellular matrix, these tissues will then fold up and close and form these sort of neural tube-like buds. And then those tissues can expand and get to be really quite large when we place these tissues in some sort of agitation. So like a spinning bioreactor or an orbital shaker, that enables these tissues to get much larger because of better nutrient and oxygen supply. And so we've also improved this method further by introducing a bit of bioengineering to enable us to control a little bit better the identities that are generated. And we're now able to quite reliably generate not only a specifically neural identity, but more specifically this part of the brain. So what's called the telencephalon, which includes the cerebral cortex, but also the choroid plexus on the inside and the ventral ganglionic eminences that will give rise to interneurons. And so what these organoids look like is something like this, where you have these large sort of lobular structures coming off. And we've injected a blue dye in here so you can see the fluid-filled cavities inside. And so just like the actual developing telencephalic vesicle of the embryonic brain, you can see these organoids form a number of these large outgrowths. And so essentially each one is kind of equivalent to one of these large lobes. And so we kind of look at them in isolation in order to understand what's going on within. So as a whole, the organoid doesn't really look like a brain, but individually we can look at each one of these lobes and we can see a lot of similarities. And I think you can see that a little bit better here, where we've sectioned through one of these organoids, and you can see these spaces here, these ventricular-like spaces. And actually, if you compare that with an actual developing cortex here from a mouse embryo, I hope you can see the similarity in terms of structure of these lobes. And then if we zoom in on the wall of these lobes, you can also see the similarity in terms of tissue architecture to that, the actual developing cortex, with these layers forming with this ventricular and subventricular zone here, which contains the progenitors. You have an intermediate zone here, which is pretty cell sparse, and then a more dense cortical plate up here where the neurons reside. And the cortical plate will then give rise to the gray matter of the brain. So I want to tell you about two short stories, one looking at the cerebrospinal fluid and its barrier, and the second examining long-range connectivity. So for the first part. So we became really interested in the choroid plexus. This is this region within the ventricles of the brain that actually generates cerebrospinal fluid. And it's actually received a lot of attention more recently because it's been shown to exhibit sort of increased flow through the brain, especially when we sleep, and it actually travels around the brain, sort of bathes the brain, and can actually travel between the blood-brain barrier and the rest of the brain, and kind of clear out waste. So essentially flow through the brain parenchyma and clear out waste byproducts, including things like aggregates. There's also certain nutrients that are being provided by the CSF. And so it's really a vital fluid for normal brain function. And there's a very large amount being produced every single day. So this tissue on the inside here has to produce quite a large amount of CSF every day. And so Laura Pellegrini in the lab became interested in developing organoids that might model this region of the brain. So she modified our cerebral organoid protocol, and she introduced a pulse of BMP4 and chiron.